kind of agencies and these kind of communication channels are still very much open. So in this project, phytoteratology, I asked myself, what it would mean if I could make a plant-human monster where I wouldn't necessarily insert human genes into the plant, but rather substitute the seed, which is like a womb, like a womb of the plant, with an artificial incubator, and that artificial incubator would have hormones of mine, so sex hormones that I would isolate from urine. So, it's not responding. Sorry for this, but what you see here are sea urchin embryos that have been treated with different concentrations of human steroid hormones. And this was an experiment I did in order to see how hormones actually did influence development. But sea urchins are nevertheless animals, right? And I was going to do something like this, or going to try to do something like this with plants. And so I isolated steroid hormones from my urine, and then I had to activate them. And I kind of, I kind of tested the urine extract on sea urchins, because endocrine disruptors, that is hormones that influence the hormonal system, have a very specific type of toxicity. It's called the non-monoclonic response. Basically, what you would usually expect with toxins is the higher the concentration, the greater the effect. But it turns out that when you have endocrine disruptors, so toxic substances or non-physiological substances that disturb the hormonal system, you will get this kind of curve where by diluting it, you will reach a certain point, which is this one-time dilution, where the effect will be there will be almost no effect, and then you dilute it further, and the effect becomes stronger. And this is because the hormones are acting on these bodies not through brute force, not like high doses of antibiotics, but really they're interfering the messaging pathways, right? So they are speaking to the internal organization of these organisms. And on that note, actually the antibiotics, as they occur in nature, also act more in the lower doses than we use them. So they kind of interfere with the languages, like they're more jamming communication than really at this dose that has to kill and sterilize that we use, which is also then thought to be the reason for so much antibiotic resistance. So is there any questions in relation to this?
So I'll ask one on behalf of the people in the audience, and also on this one to clarify. So to get the hormone, you're adding urine, yeah, and then you see this dip in the middle. Is this dip also, you think, related to what they're used to? Like it's a level that they're used to seeing somehow? Oh, can you hear me?
No.
Ah, they can hear me, but she can't. Okay, so I need to somehow match now this mess of microphones with. Okay, can you hear me now?
Yeah. Yes.
Yeah. Okay, I see. So so if we want to talk to Spella, I think my mic is here, so we need to make that directional, and then they can hear me at home. And do you know how to turn that off? No. Okay, I do. Okay. Okay, great. Yeah. Okay. So So my question was we've got a couple questions. One I'm going to field for the audience and our audience in Milan. Yeah. And then I wanted to ask quickly about the dip that you have. First of all, this is urine you put in as a way to get the hormones in. The way you're adding the hormones was adding urine, yeah? And the dip, do you think the dip is there? Sorry, I think can you hear me? Yeah? Can you hear me? Eh? Okay. Okay, now? Better?
Yeah.
Okay, so do you think that dip, you add the urine to the urchin mix, do you think the dip has to do with them being used to that amount? Like it's something that they'll see a lot of, and that's why they're not reacting so much to it?
I mean, it's actually the opposite. So
Mm.
the dip was when the dose was super effective, and then wait, no, so that they're used to. Okay, sorry. Well, no, it has to do with the way that they're reacting to the substance. So it's really complex, right? Because hormones enter like the network of communication, signaling, internal. And as you know, like almost every step has some feedback loops, right? And also it can be, so the dissociation constant of these different sensors is different, right? So what you might either, nobody really knows why this happens, but there's 4 different mechanisms that are likely, and it's like a combination of these mechanisms. So one of them is, for example, that detoxification is happening, but only that the sensitivity of the mechanisms for detoxification is a bit lower than the sensitivity for triggering the signaling cascade, which means that when you lower the dose, it will still, or actually the opposite, you lower the dose, and at one point the perturbation signaling cascade will still be significant, but the detoxification system will no longer recognize it. So it's kind of like it's kind of like this. It has to do with, also on which systems it works, agonistically or antagonistically. So it's kind of like a total mess, and that's sort of the beauty of it. And the reason why I did this preliminary experiment, even though it is kind of cruel, I had the chance to do it because I was at the moment at that time on a residency in the Marine Research Institute in Rostov. And the other reason was that I really wanted to know that, to be sure, that what I was isolating from the urine and activating, right, had some potency on the biological, the biological body. So I was using sea urchins as biosensors to know if I isolated anything interesting. And actually here you see sort of the network of, this is a metabolism of hormones in mammalian cells and the different hormones that you might expect in human urine. And this is actually a chromatogram of the samples that I sort of purified from the urine and sent off to be analyzed. So not the full spectrum of hormones are actually present in urine, but in all organisms, the hormones are kind of very, very complex mess of things. And when you interrupt or interfere with this, the outcomes are kind of unpredictable, and they change the time, right, as well. Because if you, for example, start adding testosterone to your food, you will first perhaps have this initial growth of muscle tissue as a result of the added testosterone. But after a while, your body will react and want to put itself back into balance, and it will start producing more estrogen. So that's why the bodybuilders get these boobs and stuff like women traits. because it kind of has its own set points, and it's very difficult to interfere and to manipulate, even though, on the other hand, this is how organisms that have evolved in the common semiosphere also are able to negotiate their presence, right? So there's, like, we do not have, it's not like we don't have access to communication with a plant. It's just not necessarily verbal. It's our bodies that can communicate with them and do, like every time we ingest plants, every time we're near plants, and it happens like vice versa. And there is something quite subversive, I think, that our bodies know how to do this, and our minds don't. So.
Awesome. That's fantastic. Let me just sneak in the second question from our audience. This goes back to the slide you had with your, they were HeLa cells maybe? When you had the, yeah, you had your cancer cells that were, that possibly were symbiotically ingesting a cell, and the question was just, can you clarify what it is that we were looking at? Did I recap it well, that these are the cancer cells and we're watching them in action, sort of slurping up what is from a plant, but that they're taking up as and adding to their own symbiotic system?
I didn't hear you well enough to catch
Oh
the
shit,
question.
Jesus. Okay, did you hear anything? So the question was, yeah, you got to the right slide. Sorry, this is so weird getting used to. So you got to so they just wanted a clarification. So if you want to, we're seeing cancer cells interact with the plant cells you added and taking up components, yeah?
Yeah.
Yeah, okay, clear? Yeah. You got it? You got it?
Yeah, it was nanocleopsis algae, and they were cancer cells from the carcinoma of the bladder from a woman. They were harvested in like the early 90s, and SH4 strain, I don't know. But they were donated to me by some colleagues at the Institute for Cell Biology because I asked for a really tough species of human cells, and they said that these will survive, because they're awesome. They're super fast dividing and super resilient. And the chlorella, the nanochlorosis was chosen for the same reason. So it's kind of a single-celled algae that survives high salinity. It also survives desiccation. And I actually made 3 different systems to perform this smoothing. And I'm thinking that it might be too much to just, mix them together and see what happens. But it was actually this one that had the most successful results, because ultimately this co-culture was kept in the perfect conditions for the cancer cells. So CO2 atmosphere, 37 degrees, so rich medium, etc. And it was the algae that were totally fine with this, even living in the dark, because they had everything they needed and they were surviving. And at the end, it was them surviving and the cancer cells dying. So, yeah.
So, sorry, was there any questions here? Was there any questions with you guys? Huh? Yeah? Well, do you want to, okay, how much, yeah, how much more do you have? Just so you guys do know, with the train, you're okay till like 10:30. Yeah. So there's no. Sometimes people think, oh, they got to rush for the. If you take the train, you're good for a while. But yeah. How much? We got a bit more. Yeah. Yeah. So let's let her. Yeah. We'll let her go. You got it. Okay. Cool. Okay. So we'll let you finally continue, and then we'll save the questions to the end.
I will just briefly.
No. Take the time. Take the time you need.
Anyway, this is a video that shows the process of how to make these plant-human monsters, starting with the abstraction of urine. Let's see faster. So this is how you get the essence it uses a chromatography column that separates different components of the urine based on their chemical properties. So you get something that is like a perfume, and actually it has a very distinct smell depending on whose extract it is. And it doesn't smell like fully like urine, but what I imagine would be something more of a pheromonal, yeah, pheromones basically. And here you saw the plant, Arabidopsis thaliana. So to start making these plant human babies, I take immature seeds and abort these embryos from the seeds, and then they're placed on a medium that has plant hormones. And so these plant hormones stimulate their auxins, and they sort of confuse the program of development that these plants were in, right? So before they were embryos, and then as soon as you add this synthetic oxy, which is in higher concentrations, actually used as a herbicide, they start producing this so-called callus or scar, sort of like a scar tissue, or actually you could call them undifferentiated cells or stem cells. And after you get these clumps of stem cells from these embryos, I place them in incubators, and this environment contains the extract of my hormones, right? So actually, as these new embryos, plant embryos are formed, they're influenced by this environment that contains human hormones, or specifically my hormones, right? And you might say, but how do you know that it affects them? Well, there's several reasons. I can be sure, even though this was not a scientific experiment, so I didn't do the science to statistically prove that there is an influence. Other people have done so, because recently, or I would say 15 years ago, might be recent, it was discovered that plants actually have almost all human steroid hormones in their bodies, and that they act on them in a similar manner as the plant hormones do. So the signaling, the hormonal signal is conserved. So there's progesterone receptors that they already identified, right? And they proved that estrogen, testosterone, and progesterone, and androstenedione actually act on the plant. So here we have, a very different type of conception of, making an organism in your image, if you like, right? And it's, I would say, a softer type of care, or it's still forcing, or it's still sort of molding the plant, but in a transient manner. So it affects the epigenetics, it affects the morphology. But when these plants grow up, these are the incubators, or they grow up and have seeds, these seeds will have most likely erased any memory of being brought to term, if you like, in this human environment, informed by the body of a human. So I was wondering, what kind of meaning does that have? My conclusion was actually surprising, because instead of really making plant human monsters, I realized that because I was using science as a mediator, a lot of this connection was lost, and both the plants and myself, even though we are fully capable of kind of surviving on our own and interacting and, caring for things, being part of this musicology within the context of the sterility of the scientific protocols that demand the timing, the cruelty, if you like, of performing these procedures of abortion on the plant. It seemed that I was not so much paying attention to the plant, but really dealing with the interface, and the interface in this case was the scientific methodology and scientific technique. So there was a bit of a, kind of unexpected realization coming from this project. And then this latest one that I will just briefly mention is also addressing what I spoke about, which is what artificial intelligence and algorithms are doing to the way that we see ourselves and see other organisms. It's a humorous project. It looks at the stomata, so the leaf pores. You can see here's a little setup with a microscope that looks at the leaf pores. So the leaf pores open and close in order for the plant to be able to receive and exchange gases, so oxygen, CO2 exchange, but also water. And if there's pathogens coming, it will close the leaf pores because leaf pores are like mouths. They are an opening to the inside of the plant. But in fact, if you look at the leaf pores through on a time lapse, they actually look very much like mouths. So this project was about lip reading the stomata. So this is how the computer does it. So it tries to find the opening. It's a very crude algorithm. So it's the first step. But then this is how, oops, yeah, sorry, where are we? This is how deaf people do it, because deaf people are the most well-versed in actually lip reading, which is a very challenging technique, because it's actually it has to be able to predict what the words are based on context, based on how the word would follow, and it's not straightforward. There's no way of correctly reading lips, because it's not only about the mouths. A similar mouth position can have very different sounds coming from it. So it was a project that would kind of explore the limits and the limits of communication, this imperative to communicate on one's own terms, and also to embrace the apophenia as the cognitive mistake of seeing something when that is not there, seeing faces in the clouds, etc. But it's also an artifact of all the neural networks of deep learning. This Google dream, that is basically apophenia, so recognizing certain patterns where that's not what the computer is really looking at. So yeah, that's it. Thank you for listening and inviting me.
Awesome. Cool. So, you got Percy. You got a. Oh, he's got food now. No. Okay. Good. Let me see. What I like. I mean, a lot of the time for you, you're operating on a cellular level, right? I mean, for me, I think. Tell me what you think. Oh, sorry. I forgot this. Yeah. I gotta do this shit. Sorry. So, like, for me, a lot of these projects really. No, sorry. Is it now? Yeah. No. Okay. Shit. Okay. Hopefully, I can position my laptop for you guys to. Okay. How's this? How's volume?
Okay.
Volume now. You can. You've gained the skill in the interim. I'll try and yell a bit. Is this, yeah? Are we somewhat, you can hear me a bit? Okay, I think you're gonna have to literally kind of be right by my laptop. Okay. And we don't, no, we're still gonna need these microphones. Okay. Well, are you ready now, Rosie? Yeah? Okay, so now, yeah, that's fine. Here, you want to, yeah, come. Come up here and yell in her face, like just, yeah. So this is for the audience and then yelling in her face.
Hi. Hello.
Hi.
Can you hear me?
Hi. Yeah.
All right. First off, great talk. It was very inspirational. So I have a question. I wanted to know your opinion towards bio art versus technology in general. It is my understanding that part of the mission of bio art is to question and perhaps to raise awareness for the limitations and or foundations of life forms, and the act of living. But technology nowadays, such as like social media, on the one hand, social medias and stuff, I think it's a fact that they pull us away from this this realization of living in the present, of us being an organic being on Mother Earth. On the other hand, I think more technology could lead to more means of discovery in bio art realm. So, is there an inherent paradox, perhaps in bio art's reliance on technology versus the advancement of technology?
Versus what?
Versus, let me see how to phrase it. Yeah, that was right. So, my question was,
I didn't hear it.
yeah, my question was, is there an inherent paradox of bio art's mission of bio art's reliance on technology and its mission to help people realize that we're living organisms, the significance of us being living organisms?
Yeah.
Yeah.
Okay. So I think that sort of cuts to the heart of